Good morning. Today we will start on the last part of our sex determination in Drosophila and humans topic, that is dosage compensation. When we think about dosage compensation, we need to have an idea about the composition of the sex chromosomes in organisms. Since in your syllabus we have only humans and Drosophila, so we'll restrict ourselves to these two species. In case of humans, as you know, the female has two X chromosomes, while in male there is only one X chromosome. So how is the dosage compensation done? In case of humans, what happens? One of the X chromosome of the female is randomly inactivated in every cell in the body to match the male expression of a single X. So any one, see, of these two X chromosomes, any one is randomly inactivated. Here this one has been shown to be inactivated to match the expression of the X chromosome in the males. So it is this X chromosome being expressed in case of females, and it is this X chromosome that is being expressed in case of males. But mind it, this X is inactivated, not degraded. So this is inactivated. Coming to Drosophila melanogaster, which is also a part of your syllabus. In case of Drosophila melanogaster, it has been seen that the X chromosome in the male, the transcription rate or the level of the single X chromosome in the male increases twofold in order to match the female X chromosome. So, in case of Drosophila, the male X chromosome is hyperactivated, and twofold it in fact the transcription of the single X chromosome increases twofold to match the female expression of the two X chromosomes. And this is another for information. In case of C. elegans, which is a hermaphroditic worm and is also a very well-known model to study developmental biology and genetics, for Drosophila it has been seen that the expression level of both the X chromosomes are halved, so to match the one X chromosome of the male. So this is the various strategies adopted by the various organisms to match the expression level of the X chromosome of the female with that of the male. Now coming to humans. Start with humans. In case of humans, there are basically three steps. Just a second. In case of humans, basically there are three steps to in X inactivation. The first step is the chromosome counting. Check how many X chromosome is present. Then randomly select an X chromosome for inactivation. Finally, inactivating the X chromosome. As you can see in this picture, there are two X chromosomes. The main region or the central focal point of the X chromosome, which is involved in the chromosome counting mechanism, is known as the X inactivation center. Usually, there should be two or more X inactivation center for X inactivation to occur. Now, the somatic cells of a female has a choice mechanism that determines which X chromosome is to be inactivated and which remains active. This choice is made at the X controlling element XCE, which is in the X inactivation center region. Let me see if I have that picture. This is the X inactivation picture. Just now, as you can see by this picture, this is the mammalian. This is the mammalian X chromosome. So, as I said, so there is this X controlling element which is in the X inactivation center. So, as you can see, roughly this is the X inactivation center. Within the X inactivation center, there is a gene known as XIST, capital XIST, how it's written for humans, and XIST for mice. The full form is X inactivation specific transcript, which is also located in the X inactivation region. Now, this is basically a 17 kilobase RNA, this transcript, and this XIST RNA is actually expressed from the inactive chromosome, and it targets actually the inactive chromosome. So if you look into these two pictures, let us assume that of the 2X, the X chromosome which is to be inactivated is shaded in gray, and the X chromosome which is to remain active is shaded in purple. Now, here a comparison has been shown. The X chromosome which is to be inactivated, all the genes of that chromosome are also inactivated except this one, XIST. So that is what the comparison they have shown. While in case of the X chromosome that remains activated, the Zik gene is inactivated here, but all other gene remains active. For example, they have shown an example, as you can see, with the HPRT. In case of the inactive X chromosome, this gene is inactivated in that chromosome, while in case of the active X chromosome, the HPRT gene is active in the active X chromosome. Now what happens when this XIST gene synthesizes the XIST mRNA? So as you can see, the XIST gene is transcribed into an unusually large, it's seen here, 17 kilobase RNA. That is not translated. This RNA is not translated. This RNA has a negative effect on the gene expression of this X chromosome from which it has originated. Okay. And in this case, particularly, this 17 kilobase RNA brings about heterochromatinization of the X inactive chromosome, and therefore in the process silencing the genes on that chromosome at the transcriptional level. So the XIST RNA produces this 17 kilobase RNA, and this RNA now what it does, it proceeds to bring about the heterochromatinization. So it condenses the X chromosome, the inactive X chromosome, therefore silencing all the gene. Exactly how this is an interesting phenomena, and guys who want to get into research, if you're interested into sex determination ever, so this is an area that is still open. How the XIST RNA brings about X chromosome inactivation is not fully understood. It is assumed, the next picture shows, okay, so this is the next picture. The next picture is showing, as you can see, that this is the X inactivation center. This is the would-be inactive X chromosome, and the would-be inactive X chromosome, as you can see by the picture itself, that the it's spread out, and this is the X chromosome inactivation center, and the X chromosome inactivation center now produces the XIST RNA. Now, the moment the XIST RNA comes out, as I said, the RNA targets the chromosome from which it has originated. What happens? As you can see, the XIST RNA starts coating the X from which it is transcribed, and as you can see, the coating spreads out in both directions. What it has been at the molecular level, what it has been seen is that the XIST RNA recruits a number of enzymes responsible for chromatin remodeling, famous of which are the histone deacetylase that brings about deacetylation of histone. That is, the modification is done in such a manner that the DNA becomes more condensed in its conformation. You remove the acetyl groups from the histone, resulting in a much tighter bonding between the histone molecules and the DNA wrapped around it. So one enzyme that it recruits is the enzyme that deacetylate histones, and another enzyme that it recruits are the methylase or the demethylase that induces a methylation on specific lysine residues, lysines of the histone. You know that the nucleosome is made up of histone 2, 3, 4. Okay, so you have this basic idea. So this methylase enzymes, they methylate the lysine residues on the histone 3 and histone 4. So what this RNA does, it actually remodels, it recalls the chromatin remodeling enzyme, in this case the deacetylase, the methylase, the demethylase, bringing about epigenetic modification in that chromosome. The chromatin remodeling, as you can see, the chromatin remodeling along the length of the X chromosome, inactive X chromosome, finally condenses the chromosome into a heterochromatic structure, which is known as the Barr body after the scientist who first observed it. I'll come to that. Now, another thing you should keep in mind, that the maintenance of this gene on the maintenance of gene silencing on the inactive X chromosome does not depend on this RNA. It actually depends on the epigenetic modifications that occurs when the RNA recruits the chromatin remodeling enzymes. There is another picture. This is a picture of Bruce Alberts, and another picture is also the picture of Bruce Alberts. But this is the second picture which I want to show you is of the present edition. As you can see, so this is the ZIST RNA. Sorry, this is the ZIS gene. So they have shown this is the X inactivation center. This is a picture of one chromosome, and you can see this is the ZIST gene, and the ZIST RNA is coming out from the ZIS gene, and you can see the RNA gradually spreading over, and you can see the ZIST RNA is recruiting histone modifying enzymes, and the modification continues. As a result of this modification, as you can see, the inactive X chromosome, the X chromosome is condensed, heterochromatinized, and ultimately becomes inactive. So this is what happens in case of human female. So this is another picture, as you can see, ZIST RNA. Now coming to this particular picture. This picture actually shows that which X chromosome will be selected for inactivation is a random choice. But once the choice has been made, for example, let's look at this example. This is, as you can see, that this is an early embryo, and this embryo has a red color chromosome from the father, so it's a paternal X, and a green color chromosome from the mother, which is a maternal X. Okay. Now what happens? Now this embryo divides, and with the division, randomly in case of this one, the paternal chromosome, X chromosome, is inactivated, while in case of the other cell, the maternal X, the green color, is inactivated. Now what happens? Once this initial inactivation has been selected, as you can see, inheritance of the pattern of chromosome condensation continues. So all the progenies of this cell will have their maternal X inactivated, and all the progenies of this cell will have their paternal chromosome inactivated. So the choice is random. During the process of oogenesis, however, the inactivation is removed and the Inactivated X chromosome is temporarily activated to ensure the proper process of oogenesis. So that is basically epigenetic control done there, but for you will be studying that later on in your postgraduate classes. But for now, just know that there is this random choice of, there's no particular decision that why should I choose the paternal X or the maternal X. The choice is random, and the maintenance of the X chromosome in its silent state, as you saw, is actually done by the chromatin remodeling enzymes, the histone deacetylase, the histone methylase, or the demethylase. Now coming to this picture, which was observed by long back by the Canadian geneticist Murray Burr. So you can see this is the Barr body, and it was named as the Barr body. Don't mistake the Barr body for the Barr mutation found in as a result of duplication in the 16A region in Drosophila. So there is another Barr mutation, that is BAR Barr mutation, and this is Barr body, named after the Canadian geneticist Murray Barr. Okay, so this is the Barr body she observed. And now coming to the concept of lionization, which was given in 1961 by Mary Lyon and Leonie Russell. So the basic concept of lionization is that the Lyon hypothesis actually states that the X chromosome is inactivated to give a Barr body, and this phenomena is referred as lionization. Another thing, as I said, once inactivation has occurred, all the descendant cells have the same X chromosome inactivated as their initial progenitor cells. I showed you that picture. Once a choice has been made that yes, the maternal X is inactivated, this choice stays through all the descendants. Now, this is another count of how many Barr body will be present. As we started, that the X inactivation center actually decides. When I started today's talk, I said that the 2 or more X inactivation center must be present for X inactivation to occur. So when we have 2 X inactivation center, the X2 or more. So if there is 2, we start with that. So if there is 2 X inactivation center, so you can assume that 1 X will be inactivated. So there will be 1 Barr body. In males, since there is less than 2 X inactivation center, no Barr bodies. In turn of females, again since there is 1 X inactivation center because the other is O, nullo, we have no Barr bodies. In Klinefelter, 2X inactivation center observed. There are 2X, 1 Barr body, even though the presence of Y, but there is a Barr body. See, these are the variations. Klinefelter, another variation, 2X. So 2X inactivation center, 1 Barr body. 3X, as I said, 2 or more. So remember, there will be 1 active X in the system. So we'll have 2 Barr bodies. Both the 2X will be inactivated so that finally we have 1 active X in the system. As the number of X increases, you see that there is only 1 inactive X, we are having 3 Barr bodies. 1 active X, 3 Barr bodies for this Klinefelter. Triplo X, 1 active X, 2 inactive X, 2 Barr bodies. Poly X female, 1 active X, 3 inactive X, 3 Barr bodies. Poly X female, 1 active X, 4 inactive X. So you can see 4 Barr bodies. So this is how the system proceeds. And as I said, in 1961, Lyon and Russell proposed that the inactivation of X chromosome occurs randomly, as I said again and again, in somatic cells at a point early in embryonic development, mostly during the blastocyst stage of development. And once this inactivation has occurred, all descendant cells have the same X chromosome. Now this is the, as I was saying, is This chromosome that has been inactivated does not look like other chromosomes. And chemical analysis, finally, while I was talking about the enzyme, showed that the DNA is modified by the addition of numerous methyl groups, because I said you that the RNA that is produced from that inactivase X recruits the DNA methylase, demethylase, or the DNA deacetylase enzyme. So there is DNA modification occurring there, addition of methyl groups, removal of acetyl groups. So this actually heterochromatinizes the structure, and it becomes a darkly staining structure called the bar, name given after Murray bird. As you can see, so heterochromatinization occur, I showed you in detail, and we get the bar. And this structure becomes attached to the inner surface of the nuclear membrane when it replicates out of step with other chromosomes in the cell. As you can see, the inactive X remains in this altered state in all somatic tissues, but as previously stated, in germ tissue it is reactivated, because in some cases it has been seen 2 copies of some X-linked genes are needed for the successful completion of oogenesis. So at those times it comes out, it becomes the inactivation is removed temporarily. The modifications that took place are unwind, so it's just reversed, and the X chromosome becomes again activated. Now the next picture actually shows how that so this is basically a DNA molecule which is wrapped around the histone complexes. Now when this specific category of enzymes, so here the example of histone acetylase has been shown. So these are basically enzymes that can modify the DNA externally in such a manner, either bringing about the heightened expression level of a particular gene or silencing a particular gene. Since the modification are not inherited through sequences, the modifications actually modify the structural conformation as the structural orientation of the DNA when it is wrapped around the nucleus. So these modifications are called epigenetic changes. Now this is just a schematic picture to show you how, see, this is a condensed DNA, and when I want this DNA to become a bit relaxed, then this enzymes, for example histone acetylase has been recruited, which adds acetyl groups to the lysine residues of histone tails. The moment lysine residues, acetyl group is added to the lysine residues, the lysine residues loses its positive charge, and the histone tends to move to become neutral. As a result of histone losing its positive charge, temporarily the DNA opens up, because you know that there is DNA being negative charge and histone being positive charge. There is a negative positive interaction occurring here. So this is how actually these enzymes modify the DNA epigenetically. So now if there is an enzyme that removes the histone, removes this acetylation, as you can see, if the acetyl groups are removed, so that is done. If the acetyl groups are added, they are done by an acetylase enzyme. If they are removed, it is done by an deacetylase enzyme. So when acetyl group is added, you can see the DNA in its relaxed conformation. But if we remove the acetyl group, we'll again get this condensed formation. So this is how basically, in fact, in case of X chromosome, the inactivation is done. It's just a removal of the acetyl groups and adding methyl groups. So this is what I wanted to show you, the picture of how a simple acetylation mechanism can open up a condensed conformation of the DNA to a relaxed conformation of the DNA. Okay. Now, finally coming to the last bit of our topic, that is dosage compensation in Drosophila. In case of Drosophila, the main genes bringing about heightened activity of the male X are the sex-specific lethal genes known as MLE, maleness, MSL1, male specific lethal 1, male specific lethal 2, male specific lethal 3, and males absent on the first. These genes are together collectively known as male specific lethal. Together these genes are known as male specific lethal, which produces the male specific lethal proteins. Now, if you remember the class of sex determination of Drosophila, and if you remember that in case of females, we had this early SXL protein and we had this late SXL protein. But in case of in case of female Drosophila, we had this early SXL protein and the late SXL protein. But in case of male Drosophila, there was no production of SXL protein, there was no production of the TRA protein. It directly by default synthesized a double sex DSX male protein. Okay, so if you remember that. Now coming from that concept, in females we have the SXL late protein, which is able to bind to this MSL2 protein. So this MSL2 gene, which is present in specifically in males, this sex specific lethal genes, this in case of males, this is also present in females, sorry, I shouldn't say it this way. These genes are present in both males and females. Okay. Now, in case of females, SXL late protein is there, which can bind to the transcript of the MSL2 gene. But in males, the SXL late protein is not there, as a result of which the MSL2 transcript is free. So that is the difference that is occurring. In females, the SXL late protein is there, which can bind to the RNA of the MSL2 gene. In males, there is no SXL late protein, so the transcript of MSL2 is free. In females, since the SXL late protein can bind to the transcript of the MSL2 protein, as a result of which in case of females, no translation of MSL2 RNA is occurring, and therefore the MSL2 production is blocked. But in males, since there is no SXL protein, the MSL2 RNA is generated, and this RNA is translated to produce the MSL2 protein product. So in case of females, there is no MSL2 protein. In case of males, there is MSL2 protein. The moment the MSL2 is transcribed to produce the MSL2 protein, it now forms a complex with the products of the other male specific lethal genes. So it complexes to form a product with the other male specific lethal genes, the MLE, MSL1, MSL3 and MOF. These complexes now are just like as you saw in X inactivation center, the RNA recruited certain chromatin remodeling complex. Now this complex as a whole, the MSL2 protein along with the MLE, MSL1, MSL3 and MOF, together they form the male specific lethal protein complex. This complex can bind to there is a chromatin entry site CAS on the Drosophila male X chromosome, and in a very similar manner, the mechanism which is done in X inactivation, the complexes spread over in both directions over the chromosome. This particular complex, the MOF protein, is a histone acetyl transferase. So what happens? It adds acetyl groups as it goes on spreading over the X chromosome. This particular protein, as I showed you in the earlier picture, goes on adding acetyl groups to the lysine residue of the histone moieties, as a result of which histone tends to open up from the DNA tends to open up from the histone, and the DNA becomes relaxed, and the DNA becomes accessible to the transcription machinery, and we get a twofold higher level of transcription of the X chromosome gene in males than in females. In case of females, since there is the SXL protein and as it is able to bind to the RNA of the MSL2, what happens? No MSL2 protein is produced. Since there is no MSL2 protein produced, this MSL complex cannot be formed, and if this MSL complex cannot be formed, no chromatin remodeling occurs. So we have the normal transcription level occurring in the X chromosome of the female Drosophila, while in the X chromosome of the male Drosophila, a heightened transcription, a double dose of transcription is observed because the MOF protein of the MSL complex can add acetyl molecules to the histone lysine residues, thereby opening up the DNA from the histone, uh, favoring further the transcription machinery to access the open DNA. So this is the dosage compensation part of Drosophila, and with this the topic of sex determination is complete.